Welcome to number 6 in our Drug Free Australia series on cannabis population studies. Again, we're with Dr. Stuart Reece, who is the world authority on those population studies. So thank you, Stuart.
Oh, thank you so much, Gary, for having me. I'd like to discuss today the issue of cannabis driving human aging. And we'll just talk about cannabinoid-induced aging. Now, this is one of the landmark papers in the field. You can see in the center stylized pictures of human aging there. And these are the theories of aging, which are depicted around the circle. So there's genome stability, maintenance of the genome, the telomere attrition. The telomeres are the ends of the chromosomes. And we know that as chromosomes multiply, as people age, these get shorter. Epigenetic alterations, there are changes in the epigenome that occurs. Proteins are damaged. Nutrient signaling is messed up. Mitochondria don't work properly to produce energy and do all the other things. The cells get old. The stem cells wear out and have different cellular communication. And that was a 2013 paper. Then it was followed up, 2023, the same group, world-leading group from Madrid. Again, the primary mechanisms now have been narrowed to the genome maintenance, the telomeres getting shorter, epigenetic changes, damage to proteins, and damage to the mitochondria. And mitochondrial dysfunction is also here as well. And cellular damage is part of that. Stem cell exhaustion is part of that. Chronic inflammation is part of that. And changes in the gut bacteria are part of it. So there's some of the main theories that the world leaders feel drive aging. Having said that, they're not completely separate. There's a lot of interactions, a lot of overlap between these different pathways to explain human aging. Now, so we're going to look at the indicators of cannabinoid aging, syndromic patterns of illness. There are patterns of chronic disease that are seen in old people that occur much younger in cannabis users. Cardiovascular age acceleration. We've all heard of hardening of the arteries, a classic change that occurs with aging. The hormones change with aging in both the male and the female. The mitochondria don't produce energy properly. DNA methylation changes. Neuroinflammatory mental illness, cirrhosis, hepatoinflammation, inflammation of the liver, egg and sperm changes, genotoxicity, cancer, and inheritable cancer passed to subsequent generations, genotoxicity, congenital anomalies, telomerase inhibition. The telomerase maintains the ends of the chromosomes, keeps it long enough and stops it shortening, chromosomal damage, and mortality. So many chronic health conditions are stimulated by cannabis. There's acute intoxication, mental illness, aborts the normal lifetime trajectory. It's a gateway effect to other addictions, damage to the respiratory system, the aerodigestive system, the bladder and kidneys. And then all the ones listed here are stigmata, markers of aging: heart attacks, strokes, cirrhosis in the liver, mental illness, inflammation of the brain, immune changes, hormone, fertility, changes in the reproductive tract of the male and the female, cancers, 3 of them in children or 4 of them in children, at least 10 are linked with cancers, congenital anomalies, and genotoxicity, epigenetoxicity, DNA breaks, telomere loss, and errors of mitosis and meiosis, which is where the cells divide. And these ones marked with the asterisks are all age-defining illnesses. So this is a very important paper by Phillips in 2022, looking at chronic health conditions, acute health events, and healthcare utilisation amongst adults over 50 who use cannabis, a matched cohort study. So the acute health conditions: heart attack, stroke, bronchitis, vomiting, injuries, poisonings, falls, car crash, and falls. Chronic health conditions: coronary artery disease, hypertension, that's chronic obstructive lung disease, chronic pain, behavioural health disorder, addiction, and poverty. Hospital utilisation, inpatient and outpatient visits, and emergency department visits. And when we look at this, the body mass index is not very different. That's important. Percentage of household on public housing assistance, that's important. Tobacco use, there are some differences there, and differences in drug addiction between the cases and the controls. The chronic health condition in the sample, it's 56%. In the cases, it's 67% there, 46% there, 30% comorbid addiction, 10% in the controls. Acute health event, 32%, 11% behavioural health disorder, 45%, 16%. So some of these things are much more common in the cannabis using cohort. Now, if you look at the chronic health conditions, any chronic health condition was much more common in the cases, 90%, 70%. Coronary artery disease, 23%, 6%, nearly 4 times as much. This is the adjusted odds ratio. Chronic pain was much worse in the cannabis using group. Acute health events, any health event, 68% versus 38% with an adjusted odds ratio of 2. Stroke, 10 and 0.7. Myocardial infarction, 8 and 1. Cyclic vomiting, persistent vomiting, 5 and 1. All injuries, 17% versus 4%. Motor vehicle crashes, 0.40. Falls, 5% and 0.4. Accidental overdose or poisoning, 12% versus 4%. And a lot of these changes are highly statistically significant, as you can see there. Now, hospital utilisation, inpatient, 4 times as many, outpatients 4 times as many, and emergency department visits nearly twice as many. P-values highly significant, both before and after adjustment. This is an ageing pattern of acute and chronic illness. Cardiovascular age. This paper from our group, we measured cardiovascular age in the cannabis-only patients. You see it's much higher than in the control groups, even amongst the smokers. Endocrine disruption. This is a whole monograph from NIDA, the National Institute of Drug Abuse at the NIH, looking at all these different ways the hormones in the male and female are messed up. So this is an endocrine disruptor, and that's known to be an aging change. And growth hormone, prolactin, luteinizing hormone, particularly the reproductive hormones, but other hormones as well are disrupted. And these are the references in that paper to those. And the gonads are particularly disrupted. Thyroid hormone, bone hormone. This is the hypothalamic pituitary adrenal axis, glucose and insulin are all disrupted. We looked at ovarian aging. This is in humans. This is combined opioid cannabinoid addicts, and the addicts were had a much earlier. This is a biochemical marker of menopause. Normally in young women, the LH is more the FSH, and when the FSH swaps over and becomes more the LH, that's a marker of the premenopause. So in normal women, it happens about 46. And these combined opioid cannabinoid addicts, remember the cannabis leads to other addictions such as opioids. It occurred much, much earlier, and that was a highly significant difference, 0.015. And this is important. This paper looks at ovarian failure. Now, ovarian failure is a primary cause of menopause in normal women, but when it happens prematurely, it causes menopause prematurely. And this paper showed that damage to the DNA repair mechanism, which the scientists call DDR, precipitates ovarian failure. And we know that cannabis damages the DNA and disrupts DNA repair. Mitochondria is the powerhouse of the cell, and many studies report that the cannabinoids impede and reduce mitochondrial function. And this is a schematic here showing the respiratory rate complex 1, and this is complex 2. And all these different cannabinoids reduce mitochondrial activity. You can see once you get to a certain threshold level here, there's an exponential decline in respiratory function of mitochondria in complex 1, 5 complexes in the mitochondria, 4 and 5, counting the ATPase. And there's this exponential decline with increasing concentration that we also see in some of the epidemiology. And this here shows that how the NAD levels drop, which is one of the main cofactors in the mitochondria, with age. there's this great drop with age, showing that the decline in mitochondrial function that you see acutely in the test tube and in animals with cannabinoid administration directly copies what happens to decline in age. So this is the early studies looking at THC, the effect of THC on the NAD oxidase, which is one of the main enzymes of the mitochondria in the brain and heart. And you can see there's a decline. This is percent inhibition. So this is 60%, 80%, 20%, 10% there. So it's dramatic. Here you can see the mitochondria swell, grossly swollen mitochondria here, grossly swollen and damaged due to activity of cannabinoid. Collapse of the mitochondrial potential. So the mitochondria maintain a voltage charge across their inner mitochondrial membrane because they pump protons, and some of the cannabinoids could cause a complete collapse of this potential. These two ones here, and the cell death rate with cannabichromene or THC, you can see at the higher concentrations and even the lower concentrations, there's a rate of cell death, which is due to this mitochondrial inhibition.
And cannabichromene is just one of the many, many cannabinoids, right?
That's right. That's right. Here's the same thing in the pulmonary cells, the cells that line the lung. And this is the mitochondria there. Again, marijuana. You see tobacco up here, but marijuana brings it right down. This is the ratio. This is the JC1, which is a dye which is related to the mitochondrial oxygen oxidation state, and the mitochondrial membrane collapse and ATP collapse. And you can see with the smoke exposure here, this is the ATP collapse, dramatic collapse, and this is the energy potential drops there. This is in a different lab from Los Angeles. Now, the mitochondria, we know that the CV1 and CV2 receptors are on the cell membranes of many cells, but mitochondria actually contain CV1 receptors, which you can see here. And this is an electron micrograph, and they're actually staining them here. And what they've also showed is that all the signaling apparatus, it's quite a complex endocannabinoid signaling apparatus, is also found in the outer and inner mitochondrial membrane and in the inner membrane space, so that the mitochondria signal directly with the cannabinoids. And as we said in an earlier discussion, the cannabinoids are lipid soluble, so they go through the plasma membrane and directly bind to these receptors on the mitochondria, where they inhibit their activity. And the mitochondria cluster around the nucleus normally to provide energy for all the processes of the genome and the epigenome. But when they cluster around the micronucleus, they quickly lead to rupture of the micronucleus, which is the equivalent of a genetic atomic bomb going off, and disrupts both the DNA in the lost chromosome, the isolated chromosome, which has fallen off the mitotic spindle, or the fragment which is broken off the end of a chromosome and ends up disrupting the rest of the main nuclear genome. Now, the epigenome is like switches, which turns the genes on and off, and these methylation marks, 1-carbon marks, move on and off the genes. And cannabis has been shown to increase both the hyper and hypo methylation that's on the genome. So it both increases at some spots and decreases at other spots the normal rate of DNA methylation. Now, this is a very powerful paper from the world-leading group at Harvard, the group who study aging. And this is a little bit complex, but it's worth going through in detail. This is a young mouse. This is an old mouse. This mouse has got more grey hair, and it's got a bend in its spine due to vertebral body fracture, just as we see in adult humans. Now, on the genome, we said the genome has little marks which turn the genes on and off, but those marks and the boundaries between the genes in the young animal are clearly defined, but in the old animal, the epigenetic landscape gets flattened. So you get confusion of gene expression. The normal boundaries on-off switches aren't working properly. So, and the chromatin contact, where the chromatin contacts each other, and these become factories where all the RNA is transcribed, are clearly demarcated in the young, but not in the old. And there's confusion because there's loss of the normal hills and valleys in the epigenetic landscape. And this is a key marker on the histones, which opens up for gene expression. As you can see, it's very discrete, it's very clear in the young, and it's eroded and is much reduced in the older. And this change turns the genes off. And again, it's prominent in the young and much weaker in the older. So this is their way of showing that faithful double-stranded break repair chromatin reorganization. And what this group did, they induced just 10 breaks in the DNA, 10 breaks. And that was enough to reorganize the epigenome across the whole genome and induce an accelerated aging pattern. In other words, the changes in the DNA structure, the hardware of the gene, induced changes in the software, the epigenome, which replicated the pattern of aging. And this is another paper from David Sinclair's group, the same group. And these chaps, the Yamanaka, I don't know if you know about Yamanaka, he got the 2012 Nobel Prize. Yamanaka factors for stem cell factors to induce stem cells. This is the induced stem cell factor work. And he used these three stem cell factors, OCT4, SOX2, and KLF4. And using this cocktail, they were able to change the switches and make the animals grow younger in the eye. And they had three models. They damaged the optic nerve. There was a trauma model, a glaucoma model, and a cataract model. And by re-expressing the stem cell factors to a degree in these older mice, they were able to induce healing, which only occurs in the neonatal state. So they were able to reverse this physiology of these mice to that of the newborn. And here is a crushed optic nerve, which you can see it scars, it doesn't regrow. But after stem cell treatment, there's significant regrowth into the optic nerve, which has declined. And you can see that this is the normal optic nerve DNA methylation pattern. This is after injury, it's totally different. But after treatment with stem cell factors, it's quite reminiscent of the younger one. And this is the number of regenerating axons. And this is TET2, which we said is the gene that erases and controls the epigenome. Here's glaucoma. Here, that was healed. And the mice now are able, after treatment, are able to watch the screens moving, and they can see the mice watching. They were blind before, and now they can see. So trauma, aging, and glaucoma, they're able to reverse. So if we look at the cannabis EWAS, this is an epigenome-wide association study, summary tables from this very powerful EWAS study from North Carolina. And here we have a key summary, a table summary of these are some of the main writers of the epigenome. These are some of the main erasers of the epigenome. These are key epigenetic proteins. This is telomerase, which maintains the length, the ends of the chromosomes. These are some of the key repressors of genetic function, the polycombs, and the chromatin remodelers are here. And the point is that they've all got epigenetic hits on these key proteins which control the epigenome. So we're not just damaging the epigenome, we're damaging the machinery that reads, writes, and erases the epigenome.
So just building on prior episodes, this is damage done through cannabis use.
Yeah. And this EWAS study from North Carolina was both rats and humans.
Yep.
It was validated in rats and humans. Now, this is a very powerful paper looking at epigenetic age. Using the epigenome, we can actually measure the biological age at the epigenetic level. It's called an epigenetic age, and there's different measures of this. The point is that the lifetime cannabis use was associated with greatly elevated, these are two different epigenetic ages here, 0.32, 0.27, 30% elevation at the age of 30 years, which is when they studied. This is human cannabis users. And lifetime cannabis use here in these two, this is the same sort of thing. And
so we're talking 30 years of use, and we've got around 30% over that time.
No, it's 30, age 30.
Age 30. Okay.
By age 30, they're 30% age accelerated.
All right. That does make a little bit of difference. Yes.
Right. Which is important. And this is just breaking it down. Cannabis use 9 to 12 years prior, 5 to 10 years prior, but last cannabis use in the last 4 years.
Okay, got it.
And this is showing that the very powerful effects here and the very statistically significant effects of the cannabis use, even 10 to 12 years prior. So this is a very powerful study.
Mm.
Now, cannabis is linked with many mental illnesses. This is psychotic illnesses, but the point is all of these illnesses have inflammation of the brain as a biological substrate. So if you look inside the brain of all these mental, you'll find inflammation of the brain, and that's an age-related change. Cirrhosis has been linked with cannabis, which is irreversible fibrous scarring of the liver, and that's an age-related change. And this is the paper. This was actually a paper from the American Association of Liver Diseases that's describing the association of cannabis with liver cirrhosis. Cannabinoid signaling in liver disease. We talked about all the different, some of the different cannabinoid receptors in the cell before, and there are many different ways it interacts. Cannabis has been linked with cancer in the liver, and that's what this paper is about. And it's also been linked with anti-cancer effects. But the cancer effects happen 1,000 times lower concentration than the anti-cancer effects. In other words, you get the pro-cancer effects 1,000 times lower earlier than you do the anti-cancer effects. This is an area of ongoing study, and there may be, this is an area of active research at present, there may be anti-cancer effects for cannabinoids, but at present, the pattern that we're seeing epidemiologically is that cannabis is weighing in on the pro-cancer side, not the anti-cancer side. And so this is talking about the links of cannabis with liver cancer, and this is describing some of the ways in which that occurs. Now, this is again the EWAS study from North Carolina, and this is showing activation of these key chemokines, which stimulate immunity right across the body and have been linked with aging. And this is calorie restriction. And calorie restriction is known to be linked with cancer, with aging. So by linking cannabis with these key immune stimulators and calorie restriction, it's showing that it's directly impacting the aging process, both directly through calorie restriction and immune modulation. Now, eggs and sperm, this is a gross change in the sperm. It's a normal human sperm here, and very damaged sperm, shriveled up heads, bent heads, damaged tails, multiple tails. Some of these sperm have almost three heads, or heads in 4 places, damaged sperm, as you can see there. This is sperm mitochondrial potential. This is at increasing cannabinoid concentrations. And you can see this is the mitochondrial membrane potential, the voltage charge across the membrane. This is the motility, the swimming ability of the sperm. And this is the viability, whether they're going to survive or not. And you can see the membrane potential drops, the motility drops, and the viability drops until at the highest concentration they're all taken away. So in the male, the sperm cannabinoids control seminal fluid, falling concentration. So there's a falling concentration of cannabinoids along the male reproductive tract. The mid-cycle oviduct fluid and the Graafian follicle fluid, the concentration of opioids, of cannabinoids in the oviduct, in the fallopian tube, and in the Graafian follicle in the ovary control what happens to the ovary, what happens to the oocyte, the egg cell, which determines how it interacts with the sperm. Cannabinoids also control DNA fragmentation, sperm maturation. DNA packing and repacking. We said earlier that the DNA is wrapped around histones, which it is in normal cells, but in sperm, the histones are removed and they put protamines in. And protamines allow the super packing of DNA, so it can all be packed down and scrunched down very small to fit in a sperm head. And the genome in the sperm is largely inactive. So it can all be wrapped away and tucked totally inside tightly, so that the histones that are normally inside the DNA coils have to be replaced by protamines, and this is determined and controlled by cannabinoids. So when you mess up cannabinoids, you mess up this process of dense sperm packing. DNA is nicked inside sperm normally. That's controlled by this protein, which is also sensitive to cannabinoid impaction. DNA repairs damage, protection of DNA nucleosides, and completeness of DNA packing histone protamine transition is interfered with by cannabinoids, and that's described in those two references there. So we looked at that's a ring chromosome, a chain chromosome, DNA fragments. That's a fragment of a chromosome, and that's a ring chromosome there, which is induced by cannabis. This is incomplete and disrupted, disorganized cellular division. There's a normal twofold bipolar cell division. We have chromosomes not completely separated, and this gets broken and traumatized and damaged during cell division, and all these other abnormal forms of cell division.
So just to clarify that was from cannabis use. That's not so much an aging issue. That's just a direct result of cannabis use, which will impact aging.
That is a direct result of cannabis use, but it's also a feature of aging.
Yes.
So cannabis is inducing major genotoxic changes, which copy aging, right? The scientific word is they phenocopy aging. They're not biologically old, but they become biologically old by cannabis exposure, and that's severe genotoxic damage.
Got it.
Right. So cancers we talked about at length in a separate talk, but there's lots of evidence of cannabis linked with cancer. I just referred to our early discussion. Similarly, we had an early discussion on congenital anomalies or birth defects, and so that's also noteworthy. We mentioned that telomerase is inhibited by cannabis, and there's references to both direct effect of that and which is here. So cannabis directly impedes telomerase, and it epigenetically also impedes telomerase. So telomerase preserves the length of the chromosomes. As people get older, the chromosomes get shortened. They lose a bit off the end each time because they don't replicate properly to the end. The scientists call that the end replication problem, and telomerase is nature's answer to that. So by inhibiting that, they're accelerating aging because they're accelerating telomere attrition. In other words, the nibbling away at the end of the chromosomes. Now, this is a chromosomal translocation study, and here we have the mitomycin C, which is a positive control. That's a cancer drug, a cytotoxic drug. These are two controls, saline and DMSO, which is a standard organic solvent in many labs. And the three cannabinoids, THC, cannabinol, and cannabidiol, are testing almost as strongly positive in this chromosomal translocation as this known carcinogen, mitomycin C. And here we have more chromosomal toxicity. You can see this is an abnormal cell division with bits broken off the nucleus. Again, here it's not dividing properly. There's a chromosome, a lagging chromosome has fallen off the spindle here. Here, the normal quiet cells have been transformed. This is cancerous transformation, gone from quiet there to neoplastic there, as you can see by cannabis in the lung cells. And here you can see breaks in chromosomes in all these pictures. So it's directly inducing chromosomal breaks. Here we've got ring and chain chromosomes, as we said before. Now, this is a COMET assay where the negatively charged DNA is drawn towards the anode. Here, after scientists dissolve the cell membrane so the DNA can be withdrawn, And any broken DNA will get left behind in the tail. It'll lag behind in the tail. And of course, that's exactly what happens. Here's the comet, and after exposure here to cannabidiol and cannabivarin, you can see all this broken DNA in the comet tail. We talked about the abnormalities of cell division earlier. Now, the trisomies, the epidemiology shows clearly that 3 trisomies, 21, 18, and 13, Down syndrome, Edwards syndrome, Patau syndrome, are clearly linked with cannabis. So the lengths of those 3 chromosomes, and the X chromosome also, because Turner syndrome is involved in this as well, and the European studies show that clearly, is about 400 megabases of the 3,000 megabases of human genomes, about 12%. If you look at testicular cancer has these chromosomes implicated in it. And if you add the length of those chromosomes together, you find that's 1,250 megabases, which is 41%. And that's just a paper on acute leukemia showing that these cancers, which has also been linked with acute lymphoid leukemia, also been linked with cannabis, add those chromosomes together, you get 20%. And when you add all three of those together, ALL, testicular cancer, the trisomies, and all those together, what you get is a total of 1,760 megabases, which is actually 59% of the human genome, is directly implicated by cannabis genotoxicity in terms of broken chromosomes. And of course, that's an age-related change. Then in an earlier talk, we looked at indices of severe micronuclear damage after cannabinoid exposure, control. This is cell stasis, they're not growing. This is binucleate micronuclei. So this is double micronuclei, which is an accessory nucleus, which is a genomic time bomb. This is the micronuclei themselves. This is the nuclear buds, which bud off from the nucleus. That's an age-related change. They get little blebs sort of popping off like bugs, like Mickey Mouse ears off the nucleus. Nucleoplasmic bridges. That's where we saw where the two pronuclei separate, but they're connected by a chromosome which hasn't divided properly in the mitotic process. Necrosis is cell death, and apoptosis is another form of cell death. So you can see these are all severe measures of severe genotoxic damage, which are clearly age-related. And many studies show an increase in mortality from cannabis use, which can come from many ways. They have heart attacks, they have strokes, they have cancers, they have birth defects, they have suicide, they have poor mental illness, they have poverty, they have cardiac arrest, asystole. There are many pathways to death from cannabis, and this has now been chronicled in many different scientific epidemiological studies. Just want to talk about stem cell factors. Now, stem cell factors have been used to generate induced pluripotent stem cells to reverse the aging process in several papers, which we're just talking about here. Now, the actual factors used vary from lab to lab, from study to study. But the point is you can reverse the aging process in the lab and in mice using these stem cell factors, which proves that the stem cells and the epigenetic changes that they induce cause aging, because what it means is they can make aging go faster or slower in the lab. So this tells us that we have our hand on the throttle with this genomic and epigenomic thing. And here's the thing looking at cardiomyocytes, driving them. Here's OSK, which was used by Yamanaka, OSKM, to make heart cells younger and older. And we talked about this study earlier, recovering youthful vision, and they used OCT4, SOX2, and KLF4, OSK. And here in the epigenome-wide screen from North Carolina, you can see this is OCT4, this is SOX2, that's KLF4, and that's MYC, which are the 4 Yamanaka factors knocked off at high level hits on this epigenome study from cannabis use, from prenatal cannabis use, which significantly persist after correction for multiple testing. Other stem cell factors have also been identified, and they're also strongly implicated in this EWAS screen. See, beta-catenin here is 230 hits, KIT is 185 hits, and RAS is 146 hits. These, because they stimulate cell growth, these are also carcinogens, but they're also stem cells. And the point is they're powerful, stem cell factors, also powerfully impacted by cannabinoid exposure. And as I said, they're carcinogens, so dysregulation of the system is another cause of cancer in these patients. So the bottom line from this powerful symphony of evidence, from 14 or 20 different ways, depending how you count it, the only possible conclusion is that cannabis drives aging. And from the data that we've seen, it drives it in all individuals who are exposed. It may even drive it in subsequent unexposed or not primarily exposed generations. So this is a very big deal, because if cannabis drives aging in whole populations, and whole populations are exposed by food chain, usually without their knowledge and consent, then you're talking about a holo-endemic public health disaster, really, affecting the whole community, not only the exposed, but subsequent generations. So the aging factor is important. We've talked about cannabinoid genotoxicity expressed mainly in birth defects, cancer, and aging. But aging seems to be different because it's much more common than most of the other forms of damage. Having said that, as the incidence of cannabis rises and the incidence of potent cannabis rises in the community, you'll have seen from all the exponential curves we've looked at that there's a major uptick in cancer and birth defects, and certainly, and we'll talk about this in another talk, birth defects that were rare become common because the penetration of cannabinoids into the community becomes much more common. And it's not a case of if you double the cannabinoid exposure, you double the genotox, because the exponential relationship, if you double the cannabinoid exposure, in some cases, you can go up 10 or 20-fold in terms of the damage that you see. Thank you for your attention, Gary.
Okay, and thank you, Stuart, for presenting.